Very good morning to all of you, and I welcome you to this session. Today in this session we are going to talk about fructose metabolism, and I am Dr. Niranjan Koch. So fructose as you know is a ketohexose. So how is fructose metabolized inside the body? The entry of absorption of fructose from the intestine is much lower than that of glucose, but once the fructose is inside the cell, its metabolism is much faster than that of glucose. So some of the fructose that enters into the cell may be cells are phosphorylated, are acted upon by hexokinase into to form fructose 6-phosphate in muscles and adipose tissues. But the affinity of hexokinase for fructose is very low. Therefore, this pathway is insignificant, and it becomes important only when there is a high amount of fructose in the blood. Then this hexokinase becomes active and it can converge to fructose 6-phosphate, which can then go to glycolysis. And most of the fructose is phosphorylated by fructokinase in the liver, intestine and kidney to fructose 1-phosphate. And immediately after this fructose 1-phosphate is formed, this is again cleaved into glyceraldehyde and dihydroxyacetone phosphate by fructose 1-phosphate aldolase, or we call it aldolase B. Aldolase B is an isoenzyme of aldolase. Aldolase B. Okay. So after this glyceraldehyde and dihydroxyacetone phosphates are formed, this glyceraldehyde is again phosphorylated at the third carbon to form glyceraldehyde 3-phosphate by the enzyme triose kinase, and this is an intermediate of glycolysis, so it will form pyruvate. It will undergo glycolytic process and form pyruvate. Now this is the pathway of fructose metabolism. Fructose is joining the glycolytic part at glyceraldehyde 3-phosphate. This means that this fructose metabolism bypasses the insulin dependent phosphofructokinase step. And another point I would like to mention here is the entry of fructose into the cells is insulin independent, unlike glucose, which is insulin dependent. Now fructose metabolism part we have seen. Now what is essential fructosuria? Absence of this enzyme fructokinase causes a clinical condition that is called essential fructosuria. This is asymptomatic and it is benign conditioned, and it is accidentally detected by presence of reducing sugar in the urine. And what is hereditary fructose intolerance? The deficiency of this enzyme aldolase B leads to a clinical condition that is called hereditary fructose intolerance. It is characterized by hypoglycemia. Jaundice and vomiting. So when this aldolase B is absent, there is accumulation of fructose 1-phosphate in the blood. This fructose 1-phosphate can uh, inhibit phosphorylase enzyme, which is the most important enzyme of glycogenolysis. Therefore, there will be hypoglycemia. This hypoglycemia can be explained by this inhibition of phosphorylase. Phosphorylase will be inhibited, leading to hypoglycemia. It can also inhibit aldolase and phosphohexose isomerase enzyme. Aldolase and phosphohexose isomerase enzyme. Phosphohexose isomerase enzyme, which are the enzymes of glycolysis. So the glycolysis will not proceed, ATPs will not be formed. And since the most of the inorganic phosphates will remain trapped in the form of fructose 1-phosphate, so there will be less formation of ATP. There will be less formation of ATP or impaired ATP formation. Now, so these are some of the features of hereditary fructose intolerance. I repeat once again, since this enzyme aldolase B is deficient, there will be accumulation of fructose 1-phosphate, and this fructose 1-phosphate accumulation, this can inhibit many enzymes. It can inhibit phosphorylase, leading to hypoglycemia. This is a glycogen phosphorylase, and it can inhibit aldolase and phosphohexose isomerase of glycolytic part, therefore leading to less ATP formation. Moreover, most of the inorganic phosphates will be trapped in the form of fructose 1-phosphate, therefore there will be less ATP formation. There will be less ATP formation, and therefore, since there is liver damage, jaundice will be there and vomiting. Now, why is fructose metabolized faster than glucose? As I have told, this fructose metabolism joins glycolytic pathway and glyceraldehyde 3-phosphate. That means it bypasses the phosphofructokinase. Suppose this is your glycolysis. So glucose in the first step it is converted to glucose 6-phosphate, and then it is converted to fructose 6-phosphate in a glycolysis, and then this fructose 6-phosphate is converted to fructose 1,6-bisphosphate, fructose 1,6-bisphosphate by phosphofructokinase. This enzyme is insulin dependent. So this is the third enzyme. So this is hexokinase. Okay. So this phosphofructokinase enzyme is bypassed, because after this phosphofructokinase this will be this aldolase will split into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate by your enzyme aldolase. So when fructose is metabolized, it is directly forming glyceraldehyde 3-phosphate and joins the glycolytic pathway. Therefore, it bypasses the phosphofructokinase step of glycolysis, and therefore this step is this fructose metabolism is much simpler, and therefore this pathway is much faster than that of glucose metabolism. And moreover, the entry of fructose into the cell is not insulin dependent. Here it is, this enzyme is also insulin dependent, and entry of glucose into the cell is also insulin dependent. So since it is insulin independent, this seems faster. Now, why excess consumption of fructose is hazardous to the body, both in diabetic and in non-diabetic patients, because since this pathway is here I'd like to say the fructose conversion of fructose to fructose 6-phosphate is much faster. That is, fructokinase enzyme is more efficient than aldolase. So there is accumulation of fructose 1-phosphate. I mean, there is excess of fructose 1-phosphate, and this excess of fructose 1-phosphate it can inhibit there is excess accumulation, and it can inhibit several enzymes, as well as this impairs ATP. These fructose inorganic phosphates are tied up with fructose 1-phosphate, and therefore there is impaired ATP synthesis. This leads to liver damage. And since there is excess formation of pyruvate, there will be more and more formation of lactate leading to hyperlectemia. And since there is more pyruvate, there will be more acetyl-CoA, and this will be leading to more synthesis of fatty acids, and this fatty acids will lead to more formation of triacylglycerol in the adipose tissues. And since there is this inorganic phosphates, since the inorganic phosphates are tied up with fructose, there will be abundance of ADP and AMP, and excessive ADP and AMP breakdown will lead to more formation of uric acid, that is hyperuricemia. So there will be hyperuricemia, hyperlectemia, then and liver disease. Hyperlectemia, hypertriglyceridemia. This will be hypertriglyceridemia. Okay, just see the spelling. Then hyperuricemia, hypertriglyceridemia, then hyperlectemia, and liver disease due to in the reduced ATP formation. Liver damage due to reduced ATP formation. So these are some of the explanations in relation to your fructose metabolism. Here we have seen fructose metabolic pathway, then we have seen what is essential fructosuria. We have talked about what is hereditary fructose intolerance, and we have tried to explain the symptoms: vomiting, hypoglycemia, and jaundice. And we have also seen why excess consumption of fructose is hazardous to the body. So thank you. If you like the video, please subscribe and share, and please press the bell icon so that you will get notifications of the next videos, upcoming videos. Okay, thank you and all the best.